Hello everyone, I hope you're all doing okay. You and your relatives and friends are doing okay in these challenging times. I want to give an update of our Xenopus tropicalis Desmoid tumor model. So as a quick reminder, why do we use Xenopus? So similar to zebrafish, the embryos develop externally, are easy to access, are easy to inject with compounds like CRISPR reagents. So we have very efficient genome editing in this animal. However, unlike zebrafish, Xenopus tropicalis features a diploid genome, which is highly synthetic to the human and also has a closer anatomy. So far we have three desmoid tumor models obtained in Xenopus. One is actually APC heterozygote that develops tumors, including desmoid tumors after more. It's a slow model after more than a year. Why? Because you need to get a second hit. However, we also have a fast model of 6 weeks where we have introduced mosaic mutations by injecting CRISPR reagents directly in the early embryo. And I'll come back to that later. And then finally, we also introduced CRISPR base editors to obtain activating mutations in the beta-catenin gene. And this also is a fast model that induces desmoid tumors. So the mosaic model, APC model, we have been using so far for identification of so-called dependency factors, applying a negative selection CRISPR screen. So what is the outline? So when we inject CRISPR reagents like and guide RNAs for APC, we get a mosaic mutant animal. So some cells will acquire a double mutation in the APC gene, allelic mutation in the APC gene, out of frame mutation, and this way these cells develop as a tumor because these cells are under positive selection, right? Now we're gonna go from APC injections to multiplex injections, where in addition to APC guide RNAs, we also inject guide RNAs targeting a candidate dependency factor gene. The idea is the following, that actually we are not hitting a dependency factor gene. We will sample tumors which have B-allelic inactivation in this gene. In other words, the tumor can grow in absence of its gene product. However, if we do hit a dependency factor gene, actually cells that acquire, in addition to the APC mutation, B-allelic inactivation of this dependency factor gene will never grow out as a tumor. In fact, what we do sample, if we do sample tumors, if we do see tumors, we will see that at least one allele in these tumor cells will have an in-frame mutation, right? So maintaining basically expression of a functional protein. By doing so, right, we can identify dependency genes. And this way we identified two genes, EZH2 and CREB3L1, which were never B-allelically frame-shifted, let's say, in the tumors, right? In contrast to other genes that we also screened. Now, let's go further on the EZH2. EZH2 is a compound of the polycomb-repressive complex 2. It's an epigenetic regulator. And what is interesting is that there is already an FDA-approved compound available, tazemetostat. It's used for treatment of rare aggressive cancers. And we applied this tazemetostat to animals, mosaic mutant APC mutant animals carrying desmoid tumors. So just adding the tazemetostat to the water of these animals, we observed a shrinkage or a stasis in the tumors upon a 3 weeks treatment, as all indicated here in the graph below. Now, in order to facilitate our phenotypic analysis and also to be able to follow up drug responses, we recently embarked on introducing MRI scans, so magnetic resonance imaging, in our Xenopus model. And by doing so, what we're looking here is at an APC heterozygous animal. We do a whole body imaging. This is done here in 2 scans. And we're looking here first from the back to the center part of the animal, and then a second scan from the center part to the top, the head of the animal. So this allows us to of course, observe the different organs that are present in the animal, test the stomach, liver, heart, lungs, and so forth, but also to look at abnormalities which are present in these tissues, for instance, here in the liver, and so on. Now, of course, we're going to apply this on, or integrate this with our treatment protocol. And what we do, in fact, is before the treatment, we perform an MRI scan. We do it as a metastat treatment for 3 weeks. And then we make a new MRI scan, and then we sacrifice the animal and animals that look for presence of tumors. Right? For instance, here you see this animal contained also pancreatic tumor that we were able to easily detect in the MRI image. And actually, in fact, we could go back to the pre-treatment image and actually notice that in this case, the tumor actually grew and expanded during this 3-week period. We also found abnormalities in the liver architectures of this plastic tissue. But of course, we are interested in desmoid tumors, and indeed this animal also contained desmoid tumors, for instance, here in the back muscle, that we could trace back on the MRI images. So this is after treatment, and then we went back to the images before treatment, and we noticed that the same tumors were actually larger in size before the treatment. So now EZH2 inhibition seems to shrink the tumors, but what we do not know is why they do the shrink. So actually we're not able to detect changes in proliferation, but of course these cells, tumor cells, are slowly proliferating. We do not see changes in apoptosis. So we kind of reasoned, so maybe there is a difference actually in the organization of the tumor, more particularly in the organization of the collagen, which is prominently present, of course, in these desmoid tumors. So for this, we applied microserous red staining together with polarization microscopy, and birefringence allows us to look at the organization of the collagen fibrils. And what we noticed that in tazemetostat treated animals, the tumors, the collagen fibers in the tumors were more organized than in a mock treated animal. This is quantified here. And actually, if you look back at this picture that I showed before, so before the treatment and after the treatment, you see that the tumor here has kind of smooth edges, which is, while after the treatment, these edges seem to be more contracted. There's more indentations, indicative of a contraction of the collagen extracellular matrix. So this brings me to the work plan for the next year. We want to further improve our base editor models for introducing activating mutations in the beta-catenin gene. And of course, want to use that model now also to confirm here EZH2 and CREB3L1 as dependency factors. So, and then in addition to that, we want to apply the CRISPR negative selection screen approach that I showed before for the identification for EZH2 and CREB3L1 to also look for new candidate dependency genes. And these genes we hope to obtain, or these candidates we hope to obtain from the screen, in vitro screen performed at the Broad Institute, from which you heard from Jesse Dunn. And with this, I would like to thank the people involved in this work, all the collaborators, and of course, also especially the Desmoid Tumor Research Foundation and Desmoid Tumor Foundation of Canada for their continuing support. And I thank you for listening.